Hello, everyone. I'm Greg Smith, the Curriculum Coordinator for the BioNetwork, and welcome to today's presentation entitled Cell Enumeration Techniques. Our panelist today is Ulla Dittmar, Biotechnology Professor at Pitt Community College. We're excited to have all of you meeting with us, and at this time, I'll turn it over to our panelist, Ms. Ulla Dittmar.
Good morning, everybody. I hope everybody's doing well this morning. Today, I'm going to talk about a topic that describes techniques that are used in a cell culture laboratory. These techniques are used very commonly. They are referred to as cell enumeration techniques, but a better term is actually just cell counts. So throughout the presentation, I will be talking about cells that belong into the eukaryotic region. These are cells that are 10 microns or larger. As you may know, bacteria are not enumerated in the same fashion because they are much smaller. So we'll only be talking about cells that are large enough to view microscopically without having to use a stain or something like you would if you were dealing with bacteria. If you have any questions, please, as Dr. Smith has pointed out, you can put them into the question and answer box, and I will address them at the end of the webinar. The first question, speaking of, will probably be, you may ask yourself, why do we have to perform cell counts? What is our interest in performing these enumeration techniques? And, of course, the first reason why we do cell counts is because it is a good laboratory practice. When you routinely keep cells in culture, eukaryotic cells, it is good practice to do routine counts to always be assured not just the quality of the cells, because if you're doing a visual inspection, you may see if the cell population looks happy or not, but also you get a good idea about the quantity of cells that happen to be present in a flask or in a culture that you have. The second reason why we want to perform cell counts is because you learn to subculture cells, and we try to subculture cells at appropriate cell concentrations. That means we try to keep the cells in a phase that we refer to as the log phase, which means the cells are not kept in too low numbers, but also not at too high concentration, because that often leads to depletion of medium, and then the cells may die because they don't have enough nutrients left. Another reason why we do cell counts is because if you subculture cells or passage cells, you have to do that at appropriate cell concentrations. Again, too high or too low is not a good number to keep the cells at. We may need to do accurate enumerations of cell counts, especially if we're performing some tests on cells. Picture you're culturing some maybe cancer cells, and you're trying to test to see if a chemical has an effect on the growth of the cells. As you may know, cancer cells have unlimited or unchecked growth, and so they will continue to multiply and multiply without any kind of check in place. And so suppose you wanted to test to see if your cells are responding to a chemical, a chemotherapeutic agent, for example. It is very important then to be able to enumerate the cells or count the cells to see if the chemical affects how often the cell divides and how fast the cells are growing. Finally, it's of course when you always do cell counts and you always consistently subculture your cells at proper concentrations, you get good performance, growth performance, and so you cannot do that when you culture the cells without using appropriate cell numbers. Now, there are various methods that I'm going to discuss today, and I will hopefully describe the three of the most commonly used methods. These are three methods that we do in our laboratory here at Pitt, and the students learn how to enumerate cells using these three methods. The first method is known as the Hemocytometer method. It's a completely manual method, meaning the person who's performing the cell count has to do everything from preparing the cells to loading up the Hemocytometer to looking at the cells through the microscope to actually do the calculations manually. And so it is a completely manual method. Now, we also have a piece of equipment that I will be describing that performs automated methods. The user still has to prepare the cells, still has to load them into the hemocytometer, but from then on, the salometer will be doing the work for you and give you calculations, etc., all done by the computer. The last method that we employ in our laboratory is also mainly automated, and it is a Coulter Counter method, and I will describe each one of these methods now in a little bit more detail. So you see a picture here of a hemocytometer. The hemocytometer is basically looks like a large microscope slide, and here's a picture in comparison where the hemocytometer has been loaded onto the stage of a microscope. The hemocytometer consists of actually there are two counting chambers. There's one here and there's another one here. These two counting chambers are covered up with a small glass slide. It's called a cover slip. This cover slip holds is placed on top of the hemocytometer, and when you perform a cell count, you actually load cells into these counting chambers here into these two little slits, and I'll show you that in just a second. And so you have two counting chambers that you'll be using to do the cell count on. Now, when you take this hemocytometer and place it under a microscope and use the scanning objective, or the 4X, this is what you will see through the microscope, through the eyepieces. You will actually see a grid that has been etched into the glass. These are lines that have been drawn onto the glass or etched in there. And if you look at these really careful, you'll see that there are 9 counting squares or counting areas, sort of like a tic-tac-toe grid. Okay, the next piece of important information is how do you get the cells ready for counting when you are doing a hemocytometer enumeration. Now, first thing I want to point out is we are working, or the method that I'm describing relies on the fact that these cells that we'll be counting are suspension cells. These are suspension cells because they float freely in the liquid. They are not attached to one another or to the plastic that they grow in. They're not attached to the cell culture flask, so it is very easy to mix the cells. All you have to do is shake the flask a little bit, and then the cells will be in suspension. This is totally different if you're working with cells that adhere to each other and to the plastic, because what you end up with is you have to treat these cells chemically in order to get them to release. But we are describing a method that uses these suspension cells, so they're very easy to remove from a flask. So the first thing you'll do is you take your flask and mix the cells up by rocking the flask back and forth a couple of times to make sure the cells are well mixed up and well suspended. And then the next thing is we will remove 1/2 ml or 0.5 ml of cells and transfer the cells to a small test tube. We typically use a 1 ml pipette for that, and remove 1/2 ml to put into a small microfuge tube. The next step is to add 0.5 ml or the equal volume of a stain that we call trypan blue. It's a viability stain. And the thing about trypan blue is that if you have any dead cells that are part of the culture, they will stain blue. They take up the stain and they will appear blue under the microscope, whereas viable cells that are not dead will not take up the blue, and so they will appear colorless or clear. So this is actually how we load cells onto the hemocytometer. You insert a Pasteur pipette, and by capillary action, the pipette tip fills with the cells. The cells are now loaded through capillary action into the hemocytometer. Each chamber, each of the 2 counting chambers is loaded. The liquid kind of flows directly into the hemocytometer, and one of the things, or a common mistake that is made often is that the chambers are overfilled or underfilled, and that is one of the technical aspects of loading up a hemocytometer that you have to master when you're using this kind of method. All right, now once you have loaded up your hemocytometer, you place it onto the microscope, and we typically then use the 10x or low power objective. If you use the 10x or low power objective, you can actually, this is what you'll see. The counting squares are much larger, of course, and you can see the cells as small circular circles. They're clear. They seem to be, they do have some reflectivity, so you can see them fairly well. Pay attention to this cell here with the arrows pointing at. That is a blue cell, and that is a dead cell that has taken up the trypan blue stain. Now, you focus in on your cells, and then what we do is we're using a small handheld counter. We count all the cells that are found in 4 of the corner squares of the hemocytometer. That is the method that we use in our laboratory, but that can actually differ from laboratory to laboratory. So the person who's doing cell count will add up all the cells that are found in the 4 corner squares. These are the corner squares here that are in gray, counting only the viable cells, not counting the dead cells. And then once that count has been done, you write the number down, and then you move the stage over to look at the second side of the hemocytometer to count what's in the counting chamber number 2. Now, once you have your 2 counts done, the next thing is you actually have to use a formula to calculate the number of cells per milliliter of culture. So in order to do that, you have to take the average number of cells counted. So you take viable cells you counted on side 1. Let's say there were 310 cells on side 1. You also add the number of viable cells that you counted on side 2. In this example, 322. Those 2 numbers have to be in agreement within about 10%. Otherwise, you're going to have to reload and repeat your manual count on the hemocytometer. If the cells are within 10% of one another, then you average by adding the 2 up and divide it by 2. In this case, we have an average count of 316 cells. The next thing, or part of the formula, is you have to multiply times 10,000. 10,000 is the hemocytometer conversion factor, because when we're performing a count, we're actually counting only 0.1 microliters when we're counting 1 small square. So in order to calculate how many cells there are per 1 ml, we multiply by 10,000. The final part of your of the top portion of this equation is we have to multiply by the dilution factor. The dilution factor in this instance is 2 because we're using equal volumes of cells and equal volumes of trypan blue, and therefore we have to multiply by 2 because we diluted by a factor of 2. Finally, the last part of the formula is you have to divide by the number of squares counted. The number of squares counted is 4 because we counted 4 corner squares. Sometimes when you perform a hemocytometer count, you may have to count all 9 squares because of low numbers. In that case, you would then have to divide by 9. So now I'm going to use the numbers that I have used here as an example and plug them into the formula. Again, the average number of cells was 316 multiplied times 10,000, or the hemocytometer conversion factor, times 2, which is the dilution factor, divided by 4, which is the number of squares counted, and we end up with getting a concentration of 1,580,000 cells per milliliter, and we can change that into scientific notation, 1.58 times 10 to the 6 cells per milliliter. Of course, sometimes you may not want to just know how many cells you have per milliliter. You may also want to calculate how many cells you have in a flask. And so using the number of cells per milliliter, you can easily determine the number of cells in a culture flask if you know the volume. So then you multiply the number of cells per ml times the ml, or volume of the culture. That gives you the total number of cells. Now let's move on to the next method that is we use in our laboratory and we teach students how to use, and that is using a piece of equipment called the Nexcelom Auto T4 Celometer by a company named Nexcelom Bioscience. It's basically, it looks like a metal box. It's about a foot tall. It is attached to a laptop. And another important piece to point out is there's a focus knob that you find on the side of the celometer, as well as an opening or slit that is in the front, in which you will apply the sample slide. Now, let me talk a little bit about what the Nexcelom Auto T4 Celometer is all about. First of all, it performs automated cell counts. Unlike the Hemocytometer, where you have to actually do the counting yourself, this machine does the counting for you. How does it do that? Well, it uses a computer software that has image recognition, and the computer actually learns what cells look like. It learns the sizes of cells. It learns the shape of cells, the color of cells. The only thing you really have to do as the person using the celometer is you have to focus in on the image properly. You may also use trypan blue in this method, so you can distinguish viable cells from dead cells. And again, that would be the equivalent of what you do when you do a hemocytometer count. How do you have to prepare the cells to get them ready for the celometer use? First of all, same as you do a hemocytometer enumeration, you would have to mix the cells well. Again, if they're suspension cells, that is usually no problem. It's easy to do. Then you remove half a ml of cells, transfer them to a small test tube, add half a ml of trypan blue. In this case, we use a different concentration of trypan blue. It's a little bit less than we normally do when we do a hemocytometer count. All right, I'm getting ready to show a video how we load samples into the celometer. Mix the cells up properly. With a micropipetter, you have to remove a fairly small volume, 20 microliters. And you load them into a counting chamber that comes with the celometer. These are disposable counting chambers. You have to do the loading of this counting chamber twice, so you get 2 samples loaded into the counting chamber. The next step is taking that slide, the counting chamber, and inserting it into the slit or opening in front of the celometer. When you do your second count, you have to remove the slide, turn it, and insert the other side into the opening. So you will do 2 counts. Once you have your sample slide inserted into the counting chamber, the next thing you do is on your computer, on the program, you click on Display Image. When you have clicked on Display Image, the next thing you will see on your screen is what is actually the image of the cells. At this point, you use the focus knob that is part of your Nexcelom celometer box and get the image in good focus. Good focus is when you see the cells that are looking shiny. They almost have a little bit of a yellowish cytoplasm, and they have a nice clear circle around them, a ring around them, and that's what we use as a guideline to see when the cells are in good focus. This has to be done, of course, manually, and this has to be done for every slide that you insert when you get ready to do cell counts. Now, once the machine is doing the counting for you, you get a lot of information at the end of the cell count. First of all, it will give you the actual number of cells that were counted. In this instance, you had 280 cells that were counted that were alive, 33 cells that had taken up the trypan blue stain and therefore are dead cells. The next thing that piece of information that is generated is the average size of the cell. So it takes a reading and gives you the diameter of cells in microns. The next piece of information that you get is the percent viability, and that is calculated taking the live cells and the total cells, and then you actually get how many cells percentage-wise are viable from a total number. And then finally you get two numbers that are important, the live cell concentration and the total cell concentration, and that of course includes live and dead cells. Now if you remember, our hemocytometer enumeration gave us a result of 1.58 times 10 to the 6 cells per mil. Here, using the ceilometer, we get 1.53 times 10 to the 6 cells for the same sample. So considering there's about 10% error in either methods, that's fairly accurate. That's pretty close to what we counted on the hemocytometer. All right, moving on to the next method, and this is the automated particle counter that we refer to as the Coulter Counter. Coulter Counter was first developed by somebody named Wallace Coulter in World War II. He was trying to develop an automated method to evaluate recruits that the Navy was looking for, and this Coulter counter was able to take large numbers of samples and perform a very quick count to determine the number of red blood cells that were present in these recruits, because the Navy was looking for divers during World War II. So this method has been adapted to use in clinical laboratories and is often seen in hospitals or doctors' offices. Now, the Coulter counter is really a particle counter. You take cells that are suspended in a liquid, and these cells are sucked through a small opening called the aperture. The aperture is under an electrical current, and as the particles pass through the aperture or through the opening, this electrical current changes slightly. The change occurs whenever there is a particle going through the aperture, but is also changing proportional to the size of a particle. I will explain that with this next picture. Here you have the green dots representing cells that are in a salt solution or an electrolyte solution. We use actually an electrolyte solution that comes from the company Coulter, and it's an isoton. It's called isoton. So you have these cells suspended in the electrolyte solution, and they are being drawn through the opening here, and this is circled in yellow. This is the opening called the aperture. There is a vacuum that's actually causing the cells to be sucked through into the aperture. Now, there's a constant electrical current that is applied, that flows, and the change in the current when cells are going through the aperture is proportional to the particle going through and the size of the particle. So you can actually not only get a number but also a sizing, or you can set the parameters so that the Coulter counter will only count cells or particle that are a particular size. Now, how do we have to prepare a sample to get the cells ready for Coulter counting? Again, you start off with your suspension cells. You make sure you mix them up real well, and then remove 10 microliters of sample and put them into a sample vial. Here's a picture of the sample vial, and the isoton is in the sample vial, and we're adding the 10 microliters of cells directly into the isoton. You take that sample vial, close it off, put a lid on it, invert it several times, and then you can actually place the sample. After you mix it, you place the sample onto a platform, move it in place, and press start on a keypad. Now the keypad is attached next to the Coulter counter, and from there you can do all the functions necessary to do the cell counts. Again, I have a video that shows you first the vacuum is built, then the machine gets ready to prepare. Then you see the counting performed. You can see the numbers here going up as each cell or as each particle is passing through the aperture. At the end of the count, here's a number, 7874. What we do is hit start again and perform a second count. Hopefully the two counts are kind of close, within 10%. It's usually the criterion that I use. You take the two counts, and if they're within 10%, you can average, and our average count here is 7855. Once you have calculated the average of your two counts, you have to multiply times the dilution factor. In this case, the dilution factor is 101 because we take 10 mils of the isoton and add 0.1 ml of cells, 0.01 ml of cells, and so the actual dilution factor is 101. We also have to multiply by 2 because the Coulter counter, the way we have it set, will only count 1/2 ml. So if you plug in the numbers, 77,855 times 101 times 2 gives us 1.59 times 10 to the 6 cells per mil, and that's pretty much equivalent to the previous two counts that we have performed. So in closing, I would like to compare the individual methods. And of course, one of the things that we always try to account for is how much it actually costs to do cell counts using the particular methods. So the first thing I want to look at, how much does it cost, the equipment and the supplies. And of course the Hemocytometer, as you may have guessed, is the least expensive because you really only need to purchase a Hemocytometer, a little handheld counter, and of course you need a microscope. That's probably the most expensive part, but most laboratories do already have a microscope in place. Hemocytometer is maybe $120. Now, the Coulter counter is most expensive. It costs several $1,000s, but they also are available in various grades, so you can get one that's more expensive and has more bells and whistles than the one that we're using in our laboratory. Typically, hospitals will use Coulter counters that are much more sophisticated than the particle counter that we have. The ceilometer is somewhat in between, costs about $4,000 or $5,000. The very biggest expense depends on how many cell counts you have to perform every day, would be when you have to purchase these disposable counting chambers because they can only be used once. And so if you are working in a high-volume laboratory that does a lot of cell counts, then this could run into some substantial expense when you have to constantly purchase the counting chambers. Now, when it comes to how easy the methods are to use, these are all fairly easy to use. I teach these methods to my students here at cell culture class, and they typically are very proficient after a few trials. The Hemocytometer and the ceilometer need a little bit of practice loading only because the Hemocytometer and ceilometer both rely on small volumes. It is very important that you get the proper volume loaded. You have to have a steady hand, and so that takes a little bit of practice to get good at that. Now, another question of importance may be, can electronic records be generated? Because we like to keep up with electronically, we like to keep up with the records. And so if you work in a cell culture lab where you have to perform daily counts, you may want to keep an electronic record of your individual counts. And of course, the Hemocytometer being a completely manual method, you don't have that option. You could possibly have maybe if you had a Excel spreadsheet or some sort of a computer program, you could manually enter the record into an electronic file. Now the ceilometer, this is a major advantage of using a ceilometer, because not only can you get your cell counts saved in data files, but you can also save images of the cells in image files, and so you have a permanent record available after you perform any count on the ceilometer. You can go back, look at the number of cells, you can look at the viability, you can look at the distribution of the cell, looking at the sizes, and so that may be important to understand or to look at and keep a record of. Now, the Coulter Counter in our laboratory is not attached to a computer, but of course that option is available. You can attach the Coulter Counter to a computer, and then of course you have access to electronic record keeping. Both Hemocytometer and Coulter Counter, the way we have them set up, require calculations to be performed, and these are done using a simple calculator manually. Now, let's just look at one of the pieces of equipment, the Hemocytometer. What are limitations or advantages using the Hemocytometer? I've already mentioned that it requires some practice loading because it uses a Pasteur pipette, and it also requires a steady hand that you don't over or underload the counting chambers. Another limitation is that the lowest concentration that you count is 1 cell. If you have 1 cell in 9 squares and you apply that into your formula, the lowest concentration would be 2,000 cells per milliliter. Now, if you have very high concentration, we're talking about maybe more than 2 million cells per mil, then it gets kind of tricky to be able to count all the cells. So I recommend a dilution to be performed. Then you can count the cells quicker, and the only thing you have to keep in mind then is, in order to calculate the concentration, you have to use a different conversion factor or dilution factor, because you have diluted the cells. Continue on with limitations and advantages of the Hemocytometer. Well, if you have lots of cells that are clumping up, then you can actually try to count the individual cells, number of cells that are part of a clump. And that is sometimes, if there are lots of clumps, it may be cumbersome, but at least you can get an idea as to how many clumps there are present and how many cells are present in a clump. And this is a major advantage of using a Hemocytometer, is that you get really good visual inspection of your cell population. And you can then tell if a population is ailing. You can sometimes tell if a population is starting to not look so terribly happy before the cells even take up the trypan blue stain. Now, I've cultured cells many times and for many years in the laboratory, and an immediate visual inspection can tell me immediately whether my cells are happy or whether they are starting to decline in viability. And so using the Hemocytometer's major advantage is that you actually get a good visual inspection of your cells. By comparison, if you look at the limitations and advantages of the ceilometer, first of all, the ceilometer does have a limitation as to what number of cells you can count. Somewhere between 250 and 10 million cells is when it does the best. Anything above or below is not going to give you some good numbers. So you have to have at least 250,000 cells per mil. So for very low numbers or very high numbers above 10 million, you're not going to be able to get good counts. You also have to be very careful when you pipette these very small volume, 20 microliters, into the little slide, the chamber, the counting chamber. But the ceilometer will calculate, give you numbers of cells per mil, give you viability, etc. But you also have option to allow the computer program to calculate how to subculture the cells, how to dilute the cells. So that's an advantage. You don't have to do that manually. And it can also show you how the size distribution is of the cells. So that's a major advantage because it allows you to get a lot more information and keep an electronic record of it as well. Okay, the Coulter Counter has some advantages and disadvantages. First of all, it's very fast, so you can count a lot of cells in a very short period of time. But you can actually adjust the size of the particle to be counted. So if you count cells like we use, somewhere around 10 microns in diameter, I usually set it so that it will count any size above 4 microns. The only disadvantage is then if there was a clump of cells, they would be counted as one, unless you specify that the machine would not count any large clumps. Of course, if you set the machine so it doesn't count any large clumps, then it will not count a clump of cells at all. It will also not be able to distinguish between viable and non-viable cells because it only counts particles and you don't really get a visual inspection of it. The Coulter Counter cannot tell whether a cell is viable or whether a cell is dead. Even if you use tri-pen blue, it would only give you one number, and that is total number of cells. So if you have a population of cells does not have high viability, then the Coulter Counter method would not be appropriate.
As we wrap up, please mark your calendar for our next scheduled BioForum coming on January 21st, 2014 at 10 o'clock a.m. Registration for this event is free and will be available within the next few days online at our website, ncbionetwork.org. We want to remind our audience that if you log on to ncbionetwork.org/IET, you can take advantage of our free interactive e-learning tools that we have created. These cover a wide variety of topics, and we're certain that you will find some of these that you can use. Also, if you participate in social networking, we are on Facebook. You may keep abreast of topics in the news, future events, and other interesting articles simply by liking us. Just go to facebook.com/BioNetwork. I would like to thank our panelists today, Ulla Dittmar, once again, for today's presentation. This concludes our webinar, and on behalf of all of us at the North Carolina BioNetwork, thank you for your time and participation, and have a great day.